Hi all, welcome back to Microbiology Lectures, and this time out we're going to talk about fungi. So the kingdom of fungi, again, these are just kind of brief overviews. If you want to learn a lot about fungi, you should take a course on them. There are some fungi that are really important for humans, ones that might be pathogenic, ones that can cause illness in us, but also, of course, the fungi that we consume and utilize all the time for other purposes, like the production of like beer and wine and bread, etc. Okay, so we're going to go through some of these fungi and some of the characteristics of such. Okay, so the first thing I want to kind of get across is that sometimes when we're talking about the kingdom fungi, we'll label something a mushroom, like, oh, those are mushrooms, or oh, that's a yeast. But realize that there is no such thing as like a mushroom. That's not a group of fungi. Instead, it's actually the morphological form. And so there are lots of species that can have both a mold form and a mushroom form, okay? Or a yeast form and a mold form. And but it's the same species, okay? And so keep that in mind, and we'll talk about what it means to be considered a mold or a yeast or a mushroom. Okay, so again, that just refers to the morphological form, not how we classify them. Okay, yeasts are single-celled form, and so, if it's in a single-celled form or a spore-like structure, we'd sometimes call it a yeast, but not all spores are considered yeast, because some spores, when they germinate, they form a mold. But some fungi in a single-celled structure can be a spore, and then the spore can divide and propagate more single-cell fungi, which would make them a yeast. The mold itself is mainly hyphae, and when you stack a bunch of hyphae together, we call it mycelium. Right? But it's a filamentous fungi, and we'll talk about this in a little bit. And then normally when we're talking about a mushroom, we're talking about a reproductive structure that certain types of fungi can produce. And so just keep that in mind when we're classifying organisms. We know that fungi are heterotrophs, so they're decomposers. They're extremely important decomposers. They can decompose things that a lot of other organisms can't consume. So things like cellulose. So like you eat plant material, you can't break down cellulose. You defecate that cellulose out. A fungus then can take advantage of that waste product and break down that cellulose and get energy from it. So lots of fungi fall into the category of saprotrophs. And so the saprotrophs are the group of fungi that tends to decompose or break down material that's dead or decaying material, so leaves and carcasses of organisms, this kind of stuff. This is the food source for the saprotrophs. They do so by normally utilizing either their hyphae, their filamentous cellular structures, to excrete enzymes that dissolve or break down the organic material, and then they absorb it. Or in the case of single-celled fungi, they excrete digestive enzymes out of their single cell. Again, mushrooms are reproductive structures. So the process in doing so of breaking down this decaying or dead material releases carbon dioxide and nitrogen compounds. And so a lot of times you'll have fungi forming a symbiotic relationship with other organisms. So the fungi might be breaking down this organic material, making it so the nitrogen and the carbon is available for, say, things like plants that might need to utilize it to build carbohydrates or amino acids or nucleic acids. And so you get this symbiotic relationship between the organisms where the fungi are associated with the plant and maybe even feeding off the plant a little bit because they might be getting some of the processed sugars back. So the plant might take the carbon dioxide and nitrogen, utilize it to form sugars, and then the fungi might be taking some of that back through the root system or other possibilities. It's often said that we'd be like walking on dead organisms, just tons and tons and layers of dead organisms if it wasn't for the fungi. Yes, there are a lot of decomposing organisms out there, arthropods and whatnot, but they don't do the job that fungi do. And fungi, like, they are the decomposers of the planet, the recyclers of the planet. So some are parasitic, of course. That's why they're talked often about in microbiology. One that's of great concern to wildlife biologists, especially herpetologists or individuals that study amphibians, is Dendrobatitis. This is a fungi that forms, it destroys the capillary beds in frogs in their skin, and typically gives frogs this red color in their legs, which is normally called red leg syndrome or red leg disease, but it's due to this Batrachococcytrum dendrobatitis. And so that's a mouthful for you. But that species of fungi is a real species of concern because it can affect huge bodies of water and affect lots of different amphibians. It's also not species specific, so it can be transferred from one individual to the next, one species to another species. The other thing is because fungi often can establish themselves in spores, it can lay dormant for a long time in regions. So maybe those amphibians have been removed from the region because they all died out, and then we want to come back and reestablish their population. Well, sometimes Dendrobatitis is still in the body of water, it's still in the system, and that can hamper recovery efforts. Ones that affect humans, often they're an epidermal infection, or like the yeast infection is a vaginal infection, but often these are not life-threatening. They can be painful. They can be discoloring. Some people find them very disgusting, but nonetheless, fungal infections in humans, at least the ones that are affecting humans, are typically not life-threatening. When you have life-threatening fungal interactions with humans, it's normally due to fungal diseases on plants that we eat. Okay, so things like the potato blight, or there's blights that form on wheat and other grains that we're concerned with, that if allowed to build and a human consumes that material, sometimes there's enough toxin in those in the fungi that are affecting that plant material that it can cause a toxicity in the human, and the human will die. So again, two kind of general categories when we're talking about humans. Humans normally don't die from the parasitic fungi that are consuming their body material, but rather when they consume material or eat material that might have fungal material on them, whether that be the mushroom itself or the mold or even the yeast cells, can cause devastating effects in some individuals. But one of the kind of neat systems that we have going on in the result of fungi, that for a microbiologist is maybe not as important as some of these other interactions, but we have an entire field, lichenology, or the study of lichens, that are dedicated to the interaction between two separate kingdoms. Okay, so you have the kingdom of fungi and the kingdom of protista or protozoan, that these two entities, fungi and algae, come together and form species. And these species can't survive alone, and so they normally get named based on the fungi is typically the genera, and the algae is the species. And so their interaction is so tightly linked together that when they reproduce together as a unit most often. And so they'll break off as a unit and either be washed away with water, or some float away in the wind. And because it's both the algal and the fungi unit together, they can start a new lichen individual in a new region. And it's really kind of a very unique situation. The algae is typically the photosynthetic organism, and doing photosynthesis, making carbohydrates, and the fungi is normally consuming something that they're attached to. This could be something like a tree, it could be a rock, and so these are just examples of lichen. There are a few different types of lichen: crustaceous lichen, leafy or foliage lichen, cup lichen. So there are a few different types of lichen, but they have these properties of where the fungus is actually decomposing the material that they're attached to. Rocks, so they might be taking the minerals away from the rock. The tree bark, or the tree, and they might be taking away some of the cellulose, some of the carbon away, and they're presenting that material or bringing it in. The algae has access to that material, can utilize that material to make carbohydrates, nucleic acids, the things that it needs to grow. Same thing with the fungi. And they have this unique symbiotic relationship together. Like most people assume, this is a very ancient relationship also, based on the information we have. So let's go through some characteristics of fungi. First of all, their cell walls have a material called chitin. Chitin is a polysaccharide, and it is, meaning it's a complex sugar, and it's the same polysaccharide, same complex sugar as the exoskeleton of insects. And so also you see chitin again in some other organisms, but it's a structure that is different than cellulose. So cellulose is also a complex polysaccharide, so it's a complex sugar. But chitin tends to, first of all, it has different bonds, but it tends to be a little more flexible, and so it might be stronger than cellulose, but it's less rigid than cellulose. So it has this flexibility, especially when it's first excreted. It has this flexibility which allows for it to take different shapes, and that's probably one of the benefits for like the hyphae formation of fungi, is this ability to kind of change shape, and it allows it for it to adapt to the shape of whatever it's trying to consume, so it can allow it to kind of wrap around it easier than if it was made out of cellulose, potentially. And so again, lots of things have cell walls. I mean, plants, fungi. There are some cell wall like structures in protozoan, and then of course we talked about bacteria. They have cell walls. And just normally when we talk about these microorganisms, we talk about what might be missing or what might be included in their cell wall. And so in this case it's distinct because there's no peptoglycan, but instead it has chitin. Okay? And so on top of that, it has some other components, some simpler sugars like glucan, and it has manoproteins. Okay? And so it has these scaffolding kind of structures which build support, and then they have these manoproteins, and there are lots of different types of manoproteins. But this kind of protein layer, sometimes that allows for cell communication. Sometimes it allows for things to enter, exit. Sometimes it's what creates kind of a potential toxin, an exotoxin. And so there are lots of ways at which these manoproteins work. But underneath that is a series of glucan, and then underneath that is their chitin layer, which is excreted here by these phospholipids, by specialized proteins that excrete chitin, and it can solidify, but again it's flexible, so it takes different shapes. And then probably most interestingly is fungi use a different type of sterol than, say, animals do, and then bacteria does. They use ergosterol, which is embedded in the membrane, in their phospholipid membrane, just like animals using cholesterol, just like bacteria using their own sterols. Sterols, the purpose, these fats will embed into the membrane, and it causes a little bit of rigidity, and so it doesn't flow as much. And so humans have taken advantage of some of these things. So if we're building something like a fungicide, insecticide, then maybe we build something that can break down chitin, and then you can treat both the insects and the fungi at the same time. If you're not interested in destroying animal tissue, and or killing insects, then ergostyle fungicides are kind of more important. And so like amphotericin is one that we use that kind of deteriorates the ergostyle, and it causes holes in the phospholipid to form, which then causes it to be leaky and leak out, water, nutrients, all kinds of things that maybe that organism needs, or to build new tissue, etc. Okay, so with that, we'll come back and we'll talk about some other structures of fungi next time.